Of the special senses chapter, we kind of looked at the structures and the receptors, and then we took a look at the vision or the eye anatomy. So in this part, we're going to take a look at how do we actually, take those structures and how do we turn this into our vision. Okay, but the first thing you want to do is you want to go back and you want to just review everything we learned in the first part. Okay, so you just want to go over the anatomy of the eye. And this way, when we sort of come back to the anatomy and we talk about certain structures, you're sort of familiar with sort of where those are. And again, we want to be really, really good at labeling this picture. And this is also a good one to kind of have beside you when you're going through the chapter, just so you can quickly kind of look back for a quick little reference. Okay, so here's again is the picture. And again, even if I come back to this picture, the light is going to come in through the pupil, right? It's going to travel through all of this, right? And it's basically trying to hit this back part of my eye, right, on my retina. And then we're going to talk about sort of what happens as it passes through and then once it kind of hits that retina. Okay, so again, this is the pathway coming into the eye, trying to get to the back of the eye. Then we have our pigmented layer, we have our neural layer of the retina. And again, we're trying to transmit all these impulses back to this nerve so that we can send those impulses out that optic nerve and kind of up to the brain. So here are our vision basics. The cornea and the lens focus incoming light onto the retina. Okay, once light is focused on the retina, this is where your photoreceptors are going to come into play. And we've talked about those a little bit, but we're going to come back to those and look a little more in depth here. But what we're really trying to do is we're trying to convert this light into some sort of neural signal or into that electrical impulse kind of up to the brain. Okay, so again, here is the lights coming in. It's going to hit this pigmented layer here at the back part of the retina, and then it's going to cause these impulses to be created, and they're going to get transmitted up towards these axons here, these ganglion cells, and then they're going to transmit that impulse sort of out the back of the eye along the optic nerve and then up to the brain. Okay, but we're going to look at sort of how exactly do we turn this sort of light into this electrical impulse. Okay, so first we want to look at how exactly do we focus things on the retina. So light is going to pass from the air into the eye. It's going to move sequentially through the cornea, the aqueous humor. It's going to hit the lens, go through the vitreous humor, and again, it's trying to get kind of towards that back part of the eye, right? So then it's going to pass through the entire neural layer of the retina, and then we're getting back towards our photoreceptors, and this is again where we can kind of excite those photoreceptors and sort of create this electrical impulse. Okay, knowing the pathway is always a really good question, right? So it's always a good multiple choice question to say, okay, when light enters the eye, what structures does it pass through first? So again, if you go back to that picture, it's a good one to just kind of have in your mind and have beside you and sort of think about, okay, what does that light pass through on its way back to the retina at the back of the eye? Okay, but during its passage, it's going to be bent 3 times, and this is going to create a focal point on our retina. Okay, so this is kind of a picture of the light coming in the eye, and again, it's going to get bent a couple times, and again, I'm trying to focus it on the back of the retina. So it's bent entering the cornea, and then it's bent again entering and leaving the lens. But again, the goal is to create this little focal point, okay? And then the thing that always happens a lot of times is when we think of vision is we think about, okay, what happens when people have bad vision? And this is kind of what's happening is that we just don't do a very good job at this focusing, okay? So refractive errors are kind of when you have bad vision. So they're going to occur when the shape of the eye prevents light from focusing directly on the retina. It can be for various reasons. It can just be like the shape of the eyeball, the length of the eyeball, aging of the lens. Most people are going to have one or more of these conditions that's going to lead to their sort of poor vision, okay? So if you're nearsighted, what's going to happen is the light rays are going to focus too early in the eye. If you're farsighted like me, this is where, my kids are always trying to give me things to read and they'll put it really close to me and I actually have to move it away from me so that I can kind of see properly. But what happens is that you don't have adequate focusing power and you're going to focus things, you're not going to have time to focus them before they sort of reach the retina. Okay, astigmatism is another one where you're going to get these refractive errors, but they're going to be caused by this irregularity in the shape of the person's cornea. And what happens here is they just can't focus it properly. Okay, so you end up with this blurred or this distorted image. So when you go to the eye doctor, now you kind of have some idea of, how they're using those corrective lenses to kind of correct your specific problem, right? So here's just a picture of normal vision where I'm getting proper focusing. Nearsighted focuses too early. Farsighted doesn't focus quick enough. And then astigmatism is I get this distorted focusing. Okay, and then this is how we kind of fix things, right? Where you go in and they kind of put these lenses on and they can kind of switch things. And again, the cool thing with this one is not only do your eyes have to work independently, they have to kind of work together, right? And sometimes you can do these little tests and you can see where, yeah, I can see really good on my right eye now. But sometimes when you try to combine those images, it's going to get a little more distorted too. So it's a little trickier than we kind of think. Okay, but it's amazing. I know for me, where I wear my contacts all the time or I wear my glasses all the time, sometimes you don't realize kind of how bad your vision is until you go into the eye doctor and they ask you to read that chart, and you just realize how hard of a time you kind of have. Okay, so most of us again are going to have one or more of these problems, and that's going to lead us to sort of need contacts, or it's going to lead us to where we need glasses to kind of see properly. Okay, so a functional anatomy of our photoreceptors, and again, when you're talking about photoreceptors, you're talking about the rods and the cones, and again, we're trying to stimulate them so that we can again create these impulses, and we can send that info up to the brain so that it can be analyzed. So photoreceptors are modified neurons. They're going to be arranged with their tips in the pigmented layer of the retina, and we saw that from the photo. And then our photoreceptors, the cool thing about them is that they're going to contain an elaborate array of these visual pigments, okay? And then what's going to happen is that these pigments are going to change shape as they absorb light, and again, they're going to help us to create this electrical impulse that we can send to the brain to be analyzed. So again, when we talk about photoreceptors, we're talking about the rods and the cones. And we want to know from this picture, you can see from this one that I have a lot more rods than I do cones, okay? And we want to remember that the rods are going to be for this dim light vision, so gray tones. Cones are going to be for my color, okay? But again, good to just kind of follow the pathway. And then in this picture, you can see that these little tips here of these photoreceptors are sort of embedded in this little pigmented layer, right? So when the light hits the back, hits this pigmented layer, those photoreceptors with those pigments in there, they're going to change shape and they're going to create these impulses that I can send up to this nerve. Okay, so comparing rods and cones, and we've done this a little bit already, which your rods and cones are going to have different thresholds for activation. Your rods are very, very sensitive, and this we kind of know this already too, because when you come into a dark room and you sort of adjust really, really quickly, it's amazing how much you can kind of see in that room with just a very, very limited amount of light. Okay, or like if you turn your iPhone on and you just shine a little bit of light on there, you can make things very, very, very visible. Okay, so it responds to very dim light, and it can be as little as a single photon. Best suited for night and peripheral vision. Okay, contains only a single pigment. Okay, and this is going to be because it's only again going to give us this gray tones. It doesn't give us any sort of color. Okay, and we saw that last time with the picture of the Pringles can, when we saw it with the table and the chairs where we could see the images, but we didn't know what color they were until we sort of turned those lights on. Cones are going to be very different. They're going to need bright light for activation. So low sensitivity, they're going to contain different pigments, okay, because this is going to be what's going to allow us to see things in different colors instead of those gray tones. So here is just a summary of your rods and your cones. And again, I don't necessarily have to kind of memorize this. I can just sort of think back to, okay, what do I use each for? The which one is more comes up a lot. So you just, again, you want to remember that I have way more rods than I do cones, because that comes up in the multiple choice questions all the time. So these are my visual pigments. So we said we use these to sort of create color. So my photoreceptors can translate incoming light into electrical signals using a light-absorbing molecule called retinol. Always a good multiple choice question too, is like, what molecule am I using to create these pigments? And it's going to be this retinol. Okay, so retinol is going to combine with proteins called opsins to form 4 different visual pigments, and then depending on the type of opsin that it's bound to, retinol can absorb different wavelengths of color. Okay, so cones are named by the color of light wavelengths they absorb best, and we get the blue, the green, and the red, and we all know these of, I can combine these colors and create any color that I really, really want to, but you can see where I get these different wavelengths, and they'll get their little number just to signify which one they're going to kind of show off the best. So different colors. So when we can see colors other than blue, green, and red by the activation of one or more cones at the same time. So again, it's just like kind of nursery school, right? Where we're going to paint and we're going to take those primary colors and we're going to kind of combine them to make any color that we want to. And the example that we have here is if I'm seeing something that's yellow, the way I'm going to see that is I'm going to stimulate both the red and the green cone receptors at the same time. And then if you increase it, okay, and if you say if the red cones are stimulated more, so if I'm still stimulating the red and the green, but I stimulate the red more, you want to think about, okay, if I was seeing yellow with the red and the green, and now I add more red, what color am I going to see? And it's going to be orange, okay? So we go back to that basic kind of painting and sort of remembering what colors combine to sort of form which colors here, okay? Color blindness and for this one you can kind of see the 29 here and you can see the 45, is a congenital lack of one or more cones. So basically you're missing one of your cones, so you can't combine things to form certain colors. And the lack of red-green is going to be the most common. So this is the one where you're going to look at this one and you're not going to really see the 29, you're only going to see the green dots. And then this one is less common, but this is obviously you have the blue and the purple and the things like that are not as common for people to be lacking, being able to see the 45 versus the 29. And then I always get this question too of like kind of what do people that are colorblind see? And I had a friend of mine that I worked with when I was 15, 16 that was colorblind. And it was kind of nice because we could kind of ask him questions where sometimes you feel like you're kind of insulting someone by asking them. You don't want to make people mad or insult them. But it was really nice to kind of ask him things because you could ask him just the silliest questions and he would give you like an honest answer. So we used to kind of ask him all the time and we used to kind of test him and kind of see what colors he could see. Or we worked at this amusement park like a Six Flags, and you could kind of be like, hey, what color is that person's shirt, what color is this, what color is that. And this is kind of what they're going to see, is just a lot of these green kind of shades instead of the reds and things like that. Okay, so the next thing we're going to do, this is the end of our vision part. Okay, so in the next part we're going to go over the anatomy of our hearing structures, and then again we'll do the same thing and we'll go over just the physiology of our hearing.